Okay, I think we're ready. We have a lot to cover today. I have not graded your homework, so I will do that. The next homework is due, I think, Monday at midnight as per the original schedule. It probably is not going to go up until Thursday, but it's going to be more straightforward than the last. So it'll be more short answer style questions. Oops. Okay. So our quote today was actually our quote from last time, but I don't think I put it up because we were outside, which is happiness is good health and bad memory. So we spent a lot of time talking last time about memory. I actually took your inputs and put it into slides. So those are up. We have a slide set from last time, but I'll just go through them just very briefly here today to remind us about that. But generally, and we'll get to this again in perception, this concept about happiness is divided by people in two different ways now. One is happiness sort of in the moment, like how happy are you right now based on your working memory. And then the other happiness is really a storytelling happiness. It's based about how happy you were when you were growing up, or how happy you were when you went on vacation with your friends, or you went out to the Bad Bunny concert over the weekend or something like that, if you did that. And that's really subject to your own rewriting, as you'll see. So because you don't remember everything that you did every moment that you were at some event or while you were growing up, that's the kind of happiness that a lot of people tell you you get to choose that happiness. And what they really mean by that is you can choose to some extent how you choose to remember, and whether or not you choose to remember the happy moments or you choose to remember the not so happy moments, because that past really isn't a reality that exists today in your present. So that's where that quote comes from, I think, which is happiness is good health and a bad memory. Here's sort of jokingly saying that you can forget things that that maybe didn't go your way, or that weren't good parts of your life. This is on this. These dates are already on because I put the last class on, even though there's only like 6 or 7 slides. So these are the Zoom locations for the rest of the month of March. And remember, this will come in handy if for some reason we can't do the class in here. So you have these in advance. And then the next exam isn't until April 1st. This is the exact same reading from last time. I'm just putting it up here since we didn't have the slides to show you last time. We're going to get into genes and cellular biology today. I don't expect any of us to become cellular biologists, but it's important to have at least one class on it, and a lot of the homework is going to be on it, just basic things related to cellular biology that we'll cover here, that's in the reading, and then things you can find online if you need to. I hope to get through all of this today, so I better get to it. And if we can finish memory today, then we'll start next time on perception, which is really putting everything together, putting the senses together. We'll talk about concepts, which are how we interpret senses with the things that we have stored in our memory. So by getting through memory, having gone through senses, we really are to the point where we can start putting things together for the rest of the course. Okay, and this is what I mentioned last time we were going to finish this time. Last time we talked about different aspects of memory, which I'll summarize in a short bit. But one of the things that that's covered a lot in the types of memories is that there's factual memory as well as process memory. So process memory is more how do you do something? How do you integrate something? How do you turn on a car? How do you drive a car with a clutch? You know, all those things are process memories. We talked last time examples about typing or the guitar. It's sort of a how to. I can look up on Google all day long how to play guitar and or clarinet or something I haven't played before. And I'm not really going to be sitting there and jamming out on my first session, even though there's going to be a lot that comes back. But I can go to Google and ask it, what's the capital of Texas? And it's going to tell me it's Austin. Or I can go to Google and ask it, what time is it in California right now? And it'll have adjusted for the time change and tell me, well, it's 1:37 p.m. Pacific time. So when you get to that, the fact that a lot of factual stuff is readily available, then it starts to sort of beg the question of how much of our learning experience do we want to spend learning things that even if at some point I know the capital of Wyoming, I'm probably not going to know the capital of Wyoming later on in life if I ever need to know it. I'm going to ask Google what the capital of Wyoming is. So I'm just going to give you one example. And this is one that can be full of opinions. My personal opinion is we shouldn't spend a lot of time having kids learn the capital of the 50 states. Maybe shouldn't spend a lot of time having them learn or memorize all the towns in Puerto Rico. It's just another example. In my case, I shared with you last time, linking verbs, which are a type of word that I still know today for some reason. So what I want to do now is basically spend some time just making lists like we've done before. First list is going to be, what did you memorize? So this isn't going to help you now because you've already done it. So what sorts of things did you memorize when you were going from kindergarten to the 12th grade before you got here? And then the next slide, just so you can see where we're going, is going to be, what types of things, if we didn't spend time memorizing these types of things, what should we spend that time learning instead? So, and I just want to put together some ideas here for that. Okay, so this should be easy. If you haven't forgotten everything that you memorized in kindergarten through 12th, this is just trying to put a list together of those things. Yes? Planets of our solar system. Yes? Periodic
table. I learned the entire periodic table, to which I learned half of it in 10 minutes. So I am skilled.
Okay. Is calcium bigger than potassium?
I don't remember. I'll say.
Okay, next.
The alphabet.
Alphabet. So if we take the 26 letters of the alphabet, not the Spanish version, what's the 17th letter?
That's where I started singing the song.
All right. Alphabet. Next. History is a good one. We're probably going to have some sub bullets there. So maybe give me some examples of. Okay, so European or European history. Okay, very good. So more specifically, dates, events.
here, 10th graders are supposed to take it.
Mhm.
But for example, if you go to China, and they're going to give you probably China.
Right, so maybe I'll just put here foreign to wherever you are.
Perspective of that's like wars.
Wars. Yeah, for some reason they want us to know who fought who. I don't know why that is. You say it's because history is written by the winners. I actually want to put down kings because I remember I had a history professor that just loved to make sure we knew the king of Mesopotamia or something and whatever seed were. Okay, so history. Anything else?
Civilizations.
Civilizations, okay. Presidents of the United States. Did y'all have to learn the governors of Puerto Rico or no? Yes. Multiplication tables. Grammar. Numbers are messed up here at all. 6, 6. I guess I should add here spelling and grammar. Grammar. Did you all have to write cursive? Do you know what that is? Okay. I got a C in cursive writing in the 6th grade. I just thought I'd share that with you. Okay, that's a pretty big list. There's probably a lot more things, but that's actually a pretty big list because I remember like history, a lot of history, for a whole school year in a class seemed to be remembering wars and events and dates and things like that. Geometry. So geometry, I guess you'd call them the rules maybe that you memorized? Huh? Rules, theorems. Okay. And what? Okay. All right, so let's just say that you could have your way and you were to have children and you were to have an educational system that allowed you to not worry about all the wars, but just basically said, look, we've had a lot of wars, let's not have any more. That's the final example. And so we're not going to repeat that. What would you teach instead? Huh? Programming. Yeah, very good. Good answer, by the way. Yep, sorry. What was the another?
I would teach them whether art is some easy and something that they like doing. I enjoy doing.
So like a hobby, or what do you mean?
Technical classes. Like there's some schools that give walk barber courses.
Okay, so like vocational.
Yeah.
For example, if my kid likes writing, I will like push him to keep like learning about writing.
Oh, okay. Um, It's kind of related to that. Vocational is like skills learning, which would be more like a barber or something. Yeah. Investing?
Emotional intelligence.
Emotional? No. Yeah. So maybe the 27 influence tactics. But would you want your kids to know that? So, Mom, you're just using influence tactic number 17. I'm not going to fall for it. I'm not going to wash the dishes no matter how guilty you make me feel. Any others? Yes. Sorry, a little louder, please. Oh. Excellent. Yeah, I can see that. There's a friend of mine. He's from here in Maya West, and he believes that universities are no longer needed because Google basically can answer everything. And to some extent, for some things, like he spent a lot of time on Google learning about agriculture, basically, but not from a theoretical standpoint, just in terms of, how can he make certain things grow, and when he has certain issues with his plants, um, you know, what types of plants don't need soil and all that kind of stuff. So that's a good point. You could do some self-learning of some skills and give kids the time to actually go off and figure these things out. Yeah. Uh-huh. What? Credit? Credit management? Like airline points. And I guess schools do they still, when you guys went through K through 12, did you spend a lot of time in like music or band or something like that, like a generic kind of music class? You did? Some of you did? I guess you didn't really, you would maybe have preferred that to be more custom. Is that and then language, here you learn English? Anything else? This is a pretty good list. If I was going to add to this list, I would say marketing, promotion, website presence. This is mine. I say that because it seems like I have a good friend I work with in Barcelona, and she doesn't have a college education, but she spent most of her life working in restaurants and bars and things like that. and I partnered with her. I told you guys I have a restaurant there, and she runs this restaurant. And part of what she's having to figure out is some of the things you talked about here already: money management, credit, things like that. But also just in terms of how to promote her, business. Which at first, from an engineering standpoint, I can tell you, I totally underestimated that when I used to think about it. I used to put up a website, or you put up a website and no one's going to show up to it, basically. So it seems to me that's an ever-evolving thing that having kids know how to promote would be helpful. Interpersonal skills. Some people put that into here. I'm going to put it separately because it is a bit different. I'm going to put understanding. So you hear people sometimes in English, I don't know if there's something comparable in Spanish. They say that they call it the soft skills. And as you've begun to see in this class, it doesn't, there's really some understanding behind the tools that are involved in influencing. And so I'm going to put understanding here just to separate it a little bit, because to me it's not a soft skill. I actually think learning interpersonal science is more complicated than learning most other types of sciences. There was one more, I think someone said. Yeah. Mm-hmm. Yeah, that one's a tough one because of the political part of it. But that might be an area where you could see some self-learning. I don't know how it is here, but in the States, anytime you want to talk politics, people just like go crazy. And they don't want it really in the educational system. But that could be an area where you could do some self-learning. I would love it, actually, because I like seeing both sides of an issue and that kind of stuff. I think is important for interpersonal skills and things like that. Okay, well, this is a great list. If y'all think of anything else that you feel like was not missed here, please send me a note on. Oh yeah, go ahead. Yeah, that's a really good thing. Okay. All right, next few slides, we're going to summarize what we did last time, then we're going to get into cellular biology for about 40 minutes. You'll see this both on this slide set, because I wanted to talk about it a little bit, but you'll also see it on the last slide set. And all I did was, you might remember, I was writing down things. It's pretty much how I wrote it down. There's sometimes I reordered things a little bit just to make them flow a little bit better. But there's 3 slides worth of this stuff that I suggest that you go and refresh yourself on. We talked about there being different types of memory, long-term and short-term. We indicated that there was sort of a working memory that was even shorter, which is just kind of what you're keeping track of in your current thoughts. And in a way, this is your reality. So your reality if reality exists right now somewhere, and we use our senses and the observable, sensible energies to perceive reality as best we can at any given moment in time, we're just doing it in this moment that's called working memory. So we exist for just a little bit of time. So when I have friends that get like, they really feel bad about something that happened, I try to bring them into the future and say, well, that doesn't exist anymore. Feel bad all you want, but what do you want to do about it? You know, start talking about what you want to do about it, because that doesn't exist anymore. Flip side, when I have people that dream a lot and I see they don't get anything done towards their dreams, I tell them the flip side. So, you know, you can feel happy about your dreams, but if you want to do something, like, you got to actually do that in your moments, so that when the future comes, you're going to have moments in the future that are representative of whatever it is that you're dreaming about. My favorite phrase I hear people say is, I want to have $1 million, or I want to be rich. And I said, well, everybody wants to be rich. So what's the difference between wanting and doing the things that you need to do in your working time, all these moments to get to that point, and also recognizing that you live in this moment. So you want to be, you might want to make sacrifices like you're doing right now, sitting in the classroom. But at the same time, you want to take advantage of your time and also enjoy the moments that you have. So working memory is really important. We said that memory is not an exact replica. There's an exercise you can go through, which is like, just look at anything. When you leave here, look in the parking lot, keep your eyes open, look around, and close them for a little bit. And you're going to see that whatever it is that you envision or think you remember about this scene is not going to look anything like what you're going to see when you open your eyes. And so what we remember is not only not an exact replica, but it's a very little bit. We don't store everything. Thoughts are recreated. They're created from what parts we do remember. We talked about recreated thoughts could be manipulated by someone. That was the example. We said, you know, the car smashed into the other one. You know, how far did the broken glass go? Or how fast were the cars going? That's why in courtrooms they try to object if one of the attorneys is asking a leading question. Sometimes that leading question could also influence what the person remembers. We recall our last recollection. So even though you might think that you remember something that happened when you were a little kid, which you might, but you also might remember the last time you remembered what you remembered, the last time you remembered, the time before that you remembered, the time before that you remembered what happened when you were a little kid. So if there was a memory, like in my case, I almost drowned at a beach in Florida, or at least I felt like I almost drowned, because I remember the waves turning me over. Well, that's something that every few years I remembered. So I've got a few years now, so I've probably remembered that occasion, you know, maybe 30, 40 times in my life. So how did it really happen? You know, what was really going on at that moment? I'm not really sure I could actually tell you, even though I do remember something about an event like that, at least in my mind, is a memory. Subconscious contains a lot of memories. It's how we remember how to walk, how to play the guitar was another thing we talked about last time. Reliance on what is remembered can sometimes lead us down a path that's not right. Someone mentioned, these are all things you all mentioned, so I didn't actually put anything on here that I didn't mention last time. Songs and smells can trigger memories, so we associate things we remember with songs and smells. We also associate words with things we remember. Memory creates and uses patterns. We remember facts and processes, how to do things. Habits are like memories, they're tendencies. We forget things. And perception of time is altered by memory. So if you're trying to remember, you know, what it felt like to exist over a period of an hour, it's hard to do when you're trying to remember it with a working memory that's only going to last a few seconds. So when we think about vacations, or we think about childhood, because we're not remembering every second of every day, sometimes we can sort of tend to compress time. And this is the last slide. It's just different types of things, concepts, events, people's names, signs, what we think we felt about something, even if we're not exactly feeling that thing at the moment. People, attributes, senses, and language. Okay. Everybody, I assume, who did not take a basic biology class in high school? You did not in high school, or an advanced? Okay. Did you take a biology class here? Okay, good. Well, I'm going to show you a very little bit of biology. What I would suggest that you do if you've never had a biology class is just go online and see, like, how does DNA work, or what's the cell? The conicotomy is one we've used before. I don't have any assigned stuff, because what we're going to do is fairly intro level, but KAHN Academy has a bunch of videos that are pretty good. And in the span of like 30 minutes, I'll basically go through what you need to know here. Remember that scientists do not know now how we actually remember things. What I'm going to be showing you here, which is what I think is important for this class, is we've already talked, when we talked about digital and analog memories, electronic memories, we talked about two very different fundamental ways of remembering things in electronic systems. Those ways can exist in biological systems. And I'm going to show you enough about how the cell works and what it's made up of so that you can actually see how you could use different parts of the cell to remember things. I am not saying that this is how the cells remember things. I have my own ideas. Can't prove anything right now. But you can convince yourself, I think, and this is what I am saying is fact, that you can store information in different ways. So that's really what this is about. This is a model of the brain. And the brain is basically, depending on how you want to describe it, you've seen functional representations of the brain before. This is just showing it from a four main system standpoint, which is the cerebrum, which is most of the brain, which is where most of your consciousness and the perceptions of senses and all this stuff is going on. The cerebellum, which is where most of your motor controls activity is going on. I can't even pronounce that. Diencephalon, which is like this little, it's not labeled there, but it's this little part right here that sort of connects the brainstem to the rest of the brain. Really what we're going to talk about here is, well, if most of your processing is going on in the cerebrum, and that's where you're perceiving what you're touching and what you're seeing, what you're hearing, and your working memory and your perception of reality. Well, what's that made of, right? Because whatever that's made up is presumably what's doing your thinking, your signal processing, and what's storing the information that you're using so that you can speak and calculate things and remember the periodic table and that sort of stuff. Mm-hmm. So the answer is that it's made up of cells. It's made up of a bunch of cells. In preparing for the class, I wanted to be accurate. So I'm going to be accurate and tell you no one really knows exactly how many cells. But if you go back and look in the literature, the numbers vary actually fairly widely, depending on who's counting and how they're counting. For obvious reasons, when we look at human brains, they're not completely, but for the most part, to look at most of the brain, especially the inside of the brain, it's got to be a brain that's from a person that's dead. So by definition, the brain's dead. So it's kind of hard to go in and figure out exactly what everything is made out of inside. There are some cases where you can study a person's brain while they're still alive if you're doing some surgeries or implanting some things into the brain. But there you're only really seeing visually the outside portion. And then, of course, there are things like resonance imaging and things like that that can tell you some things about what's going on in different parts of the brain. But these numbers are, I think, fairly accurate because I put a squiggle in front of them, which means approximately 10 to 11 neurons, which is around 100 billion. Right now, the consensus tends to be leaning towards a number of somewhere around 80 to 90 billion. To me, it's the same number, 80, 90, 85 billion, 100 billion. It's a lot of neurons. Not really an incredible amount of neurons. If each neuron were to store 1 bit of information, and you had 80 billion neurons, divide 80 billion neurons by 8, you get 10 billion neurons, and 10 billion is 10 giga. So you'd have like 10 gigabytes of memory, basically, if every neuron stored a bit. And I started to think, is 10 gigabytes of memory like enough for me? Like, do I actually know more than 10 gigabytes of stuff? And for reference, like probably every single cell phone you guys have, even if you've got a flip one, not the flip one, but has more than 10 gigabytes. It's going to have probably 16 by now or 64 gigs. So I started doing a little bit of math. And I thought, well, I know a lot of words. So how many words are in the English language? Somewhere between 100 and 200,000 words. So if I knew all the words, and there was 200,000 words, and each word on average had 10 letters in it, well, that'd be 200,000. That'd be 2 million. I'd need 2 million characters, right? So that'd be, I'm just going to divide by 10 to make the math easy. So that'd be, well, no, that'd be basically 10 megabytes of information that because a character is roughly a byte of information if you use ASCII or something to encode a character. So 2 megabytes to store all the words that I use, right? And then I need a little more memory to remember how to combine the words to make sentences and stuff, right? But I don't have to remember every combination of words possible. So 2 megabytes, so that leaves me 10 gigabytes minus 2 megabytes, which is still pretty much 10 gigabytes, or almost 10 gigabytes of information. Then I thought, well, but if I take a picture here in a camera, that's going to store, if I compress it, it's going to store some tens of kilobytes of information. And surely I remember a lot more than that, right? But then I did the experiment where I closed my eyes. And I remember a few of the people that are actually sitting over here because he memorized the periodic table. Over here because he's in the front row. Over here because he almost always sits over there. Back there because I remember he was yawning a little bit more. And that's pretty much it. But even though I remember these people, like I don't have a vivid image of them. I don't have a vivid image of them. It's a faint image. And even though I've seen you every day of class, I don't actually have to restore you every day of class. I can remember kind of what you look like one time. And I'm probably not storing, however many times I could see you every second over the next hour and 15 minutes for every day we see each other in class. I don't think so. I think I probably remember like some vague image of you once and maybe twice or 3 times in case some memory cells get damaged. And that's it. So I actually haven't convinced myself that I know more than 10 gigabytes worth of stuff. If you guys figure out a way to do the math and figure out more, let me know. What you'll see in the literature is people think that the human brain can store petabytes of all sorts of information, like the whole entire internet could be stored in your brain. I don't really see that. And if it's possible, I'm not sure why, but I don't actually think I know. I think I know I think I remember a very small amount of information. I think I remember a very little amount, remember a lot of sort of rules, and then somehow I like put together these concepts when I see people. That's what I think. That's just what I think. So there's neurons. Neurons get all the fame. We talked about maybe 80 billion neurons. They get them because we know that they're connected to each other and they send electrical signals. And if you're in the computer world, you like it when electrical signals are being sent. There's another cell that's actually fairly well associated with neurons that does not get very much fame. That's called the glia. There's 3 types of glia cells, and there's also around 100 billion of them. But lately, if you look in the literature, people are starting to think that maybe these are involved some way in memory as well. They do provide support functions to nerve cells. They help repair cells, and there's 3 different types. This is a neuron, simplistic view. It's a cell. Like you have cells throughout your body. The difference is these neurons look more like, and you have neurons that are also throughout your body, but neurons in your brain are highly associated with each other in 3 dimensions. So one neuron can send through its axon and these nerve endings, and through the reception through these dendrites, and what looks like sort of a tree or a bush with a lot of limbs, could interact directly with up to 1,000 other neurons. So, so one memory cell or a processing cell or whatever the combination of neurons do, because we know they process information too, can interact directly at the same time with lots of neurons or with specific neurons. So the neuron basically has this area here. They're called dendrites. We'll talk more about dendrites in a little bit. It has the cell body here, which is called a soma, which has the nucleus inside. And then it has this axon. Not all neurons actually have this long axon. Some of them are sort of more dendrite oriented. So this axon has this another cell on top of it that basically shields it. And the idea is if you send an electrical signal, it can make its way further away from the neuron by having it go through this axon without that signal interacting with its environment. So this is like an electrical shield. Okay, so I'm not saying that dendrites store information in their 3-dimensional geometry, but I am saying they could. Okay, and I'm going to explain what I mean by this in a couple of slides. This is a 3D printed neuron. If you don't have a project and you want a project, you could consider doing something along these lines. We talked last time about how let me take that back. So, so this is a 3D printed neuron, and so what information do you use to print it? How do you decide for the dendrites which direction they're going to go in, when there's going to be limbs, how long they're going to be, which direction the limbs are facing? You could decide those things. So what I'm going to show you here, I put this together here to show you that you could decide when you're creating this very long sort of tree-like architecture with lots of branches and different smaller limbs and stuff, that if you're going from a little piece of your branch to another little piece of your branch, you could decide that you're going to rotate this 30 degrees. So you're just going to have a branch that's going to rotate this way 30 degrees. Or maybe you're going to rotate it 60 degrees. You could decide that if you're going to rotate it 30 degrees, that's going to be a 0. And if you're going to rotate it 60 degrees, that's going to be a 1. So if I wanted to store information in a dendrite as 0 and 1s, then at any given length along any given limb, I could just simply make a little bend in it. Okay? The more I bend it, it's a 1. Bend it a little bit, it's a 0. But I don't have to bend it that way. I could bend it that way. Right? So if I bend it this way, 30 degrees, then it still can look, it's still 30 degrees. But now when I look at it this way, I'm getting different answers as well. And here I said 30, but really I did it in the picture at 45. So what this picture here is, it's showing is basically saying, if I'm creating a limb, a physical structure, that this layer I can go up, this layer I can bend it 30 and call it a 0, and this way 60 more and call it, or 60 total and call it 1. And then I can take this and orient it in 3-dimensional space, 45-degree increments, and then basically here get I can count up to 0, 1, 2, 3, 4, 5, 6, 7. Right? And if I was doing it at the 30-degree position, I could get me 7, 0, 7. And then if I just went to 30 degrees more to the 60 degree, then I could count 8, 9, 10, 11, 12, 13, 14. So I can store 2 bytes of information just in one little segment of a limb. So if I had a file that was like a photo of me in bitmap that was comprised of a bunch of 0s and 1s for each pixel in that image, then I could potentially make a 3-dimensional structure that was effectively contained the same amount of information. Now how you would read that out, I don't actually really know, but it'd be cool, something art for your wall. So the point is that you can use physical orientation to store information. Okay, I told you that there's other cells besides the neurons, and there's about as many of them, and they're called glia. One of the glia is actually getting a lot of attention recently. It's called an astrocyte, because there's some evidence that it's involved in memory. And there's 4 bullet points here. I'll go through them. It's a little confusing. I tried to make it easier to understand, but I failed. So memory formation during learning requires some suppression of retrieval of past memories to avoid interference. And there's this a molecule called acetylcholine that's found in animals when learning is going on. And then the astrocyte, that's the cells that are throughout your brain that are associated with the neurons. It releases a neurotransmitter called glutamate. And in the end, basically, these things interact. So it converts this, this chemical here into this glutamate associated compound that then is involved in learning. Again, we're not going to be experts in cellular biology. The point I'm trying to show here is that you can have one cell, what one cell is doing, influence what another cell is doing, so that if what you remembered was in one cell, you could use that to modify the behavior of the other cell. So that would be comparable to saying, okay, well, maybe neurons are just communication channels, and maybe the real thinking or really the memory is going on in these other cells, and they're modulating these communication channels. Maybe that's what's going on. And now I'm going to go into some specific examples how this modulation can occur. So, so the cell basically has a membrane. I should probably show you a picture of a cell right now. But a cell you're probably familiar with is the egg, like a chicken egg. So a chicken egg's like a cell, basically. The nucleus is usually attached to the yolk. It's a really big cell. But the cell has a membrane that protects and keeps the liquid inside. Right? So the cells in your brain don't have hard shells like the eggshell does. But even if you take off the eggshell underneath, there's like a little membrane that kind of keeps the egg together. That's why you can break it and put it on a surface and it doesn't just completely go. It kind of stays together until you pop it. So those membranes basically allow things to go in and out of the membrane through things called proteins. So these are proteins, and this is what's called a channel protein. And it's what allows something to go from inside to outside the cell. This is really important for neurons and electrical signals. I'll show you how electrical signals are generated based on this, on proteins that go through the membrane. And this is how that happens. So you've heard of neurotransmitters before because we've mentioned them in class. Neurotransmitters are basically chemicals that activate neurons and allow them to send electrical signal. So this is that same chemical we talked about a little bit a while ago. You see this ACH here is the same acetylcholine here. So if another cell is emitting acetylcholine, that acetylcholine can go to a neuron cell, which is a nerve cell, and bind to this protein and basically cause this protein to open up. And when it opens up, you remember from the memorization of the periodic table that you have these sodium ions up here. And these sodium ions are in a much, much higher concentration outside of the neuron than they are inside of the neuron. So when it opens up, they come pouring in. They come pouring in not because of any sort of being driven by current. They come pouring in because there's just a bunch more of them. So it's like a diffusion process. So, but when they come in, because they're charged, when they come in here, and by the way, you have some potassium ions also going out. You might remember potassium is bigger than sodium, so they don't really quite go out as fast. When you have all these sodiums coming in because they're charged, they build up a lot of charge in the cell. And that charge in the cell changes the voltage of the cell. And so you have other kinds of channels, which are voltage threshold protein channels that open up. And those allow the potassium that I told you about that's inside the cell to go out of the cell. Okay. So you have just these little neurotransmitters going about that are released by someone that wants something to happen. They touch that protein, it opens up, a bunch of sodium comes in, so much so that it changes the voltage, which then opens up other channels that allows a bunch of potassium to come in. And why do we care? Well, we care because that's what we've been talking about for weeks now in class is an action potential. That's what is the electrical signal that's allowing information to go from one part of your body to another part of your body, or from one part of your brain to another part of your brain, or from one part of your brain, a neuron, to another adjacent neuron or one that's connected by way of the axon to another one or through its dendrites to other neurons. So this is the information that's basically flowing over long distances in your brain and throughout your body. If you modulate electrical signals, then you can transmit information. And this is the case with your cellular phone, with displays. Basically, it's the case with almost everything we do in computing and electronics and communications. So how do we modulate? We modulate with memory. So it could be that the memory processes are in other cells, and that those other cells, when they want to influence and communicate what they know, they modulate the neurotransmitters by sending out ACH to them to cause them to act. Okay, now we're going to talk about cells. I'm doing good. We got about 20 minutes left, and that's about as much as I want to go into cellular biology. All right, so this is a cell. This could be any cell. I haven't drawn it as a neuron because the neuron cell you're going to see has a characteristic dendrites and axons. But it's a cell. It has a nucleus just like the neuron does. And it's got this liquid. Most of the cell is just made of liquid, mostly just made of water. And in there it's got a bunch of chemicals. We already talked about that it has a lot of potassium inside. That outside there's normally fluids that have sodium. It's got a membrane like that egg yolk, or like the egg yolk, and also like the egg clarita that's got the membrane that kind of keeps the liquid inside. And it's got a lot of things. One of the things it has that's really important is the nucleus. We're going to dive into the nucleus and talk about things going on in the nucleus. And then we're also going to talk about these ribosomes. We're not going to talk about any of these other things here. Not because they're not important, but because my point will be gotten across just by talking about what most people consider to be the most important parts of the cell. The cytoplasm is basically everything that is the liquid plus the things that are floating around inside the cell. So if this was a chicken egg we were talking about, it would be what's inside the clarita plus anything that's floating around inside the clarita. It's not the nucleus. The nucleus is considered separate. Cytosol is the liquid. So cytosol is a part of cytoplasma, but cytosol doesn't include the things that could be floating around in the liquid. So why is this important? It's important because we talked about before, remember all those sensors we had, whether it was touch or sound. You had receptors on the cochlea, you had receptors at the back of your eye, you had receptors in your nose, on your tongue, and the taste buds. Those receptors, when they do things, they're going to cause chemicals to flow and ion currents to flow through this cytosol that allows it to interact with other things that are inside of the cell. So the cytoplasmic, so the membrane kind of keeps everything together, and inside you have the cytosol that allows chemicals to move around, molecules to move around in a liquid. The nucleus. Nucleus contains the majority of the cell's genetic material. When I'm talking about genetic material, I'm basically talking about DNA. And those DNA molecules, along with the proteins, form what's called chromosomes. So if you've taken biology before, but not recently, all this probably sounds familiar, but you may not remember exactly what it does. So we'll start with the chromosomes. These are the chromosomes. You have 46 of them in most of your cells. And they, for the most part, contain the same DNA. It used to be that people thought that the DNA was exactly the same, like in all of your cells. Now, most scientists that are involved in this area don't actually think they're exactly the same. They think that they're actually different, but just a little bit different. And unless they get to be a lot bit different, where it affects how the DNA replicates itself or how genes express themselves, then it's kind of okay. Some people think that maybe those little bit of differences, a base pair here, base pair there, could be how memories are stored, especially long-term. Because DNA is a good way of storing information for a long time, since the DNA that you're born with, the sequence for most of it, is pretty much the same throughout your body for all of your life. If you are able to store some memory in your DNA for long periods of time, then it probably means that you're passing some of that memory to your kids. Because when you have children, generally you're going to donate your DNA, your partner's going to donate their DNA, and it's going to combine to make, well, they're going to have different chromosomes going to combine to be that person. So you can pass DNA off. So to the extent that you have any memory within DNA, you could actually think of a baby being born with memories already. And then add into those memories whatever it is that they get on their own from living and having all the senses of the reality that's around them. Okay, a lot of information on the slide. I suggest you go through it afterwards. Short person, I think I mentioned all these things pretty much for the most part. There are 4 molecules that are called bases that make up DNA, and that's what encodes your DNA. They pair up. A pairs up with T, C pairs up with G, and you can have up to 3 billion bases. So you should think of, if a base is one A molecule paired up with a T molecule, they always pair up either AT, TA, CGGC. So all of your DNA, which is spread across the chromosomes all together, and they're not all connected, but all together have around 3 billion bases. So if you divide that by 46, then you get, I guess, something on the order of 100 million bases in one strand of DNA. Only a very small part of that is actually what you hear of as the genes. So, you hear that genes do a lot. They basically create proteins. They decide what happens in your cell, but it's just a very small percentage of the DNA actually is made up of genes. The other 99%, people used to think was junk, but scientists now are figuring out that it's not actually junk necessarily. It just doesn't code. It does other things. It may facilitate coding. It may inhibit coding. So a lot of things in DNA, we don't know what it does. So now I want to show you, I'm going to bring you back to the world of computer science for a second. Because if we know that DNA starts with you, pretty much stays the same throughout your whole life, you can pass it on to your descendants. And only 1% is involved in genes. What's the other 99% involved with? We know that you can make small changes to the bases. You can change a base here and there, and it's not going to mess you up. But maybe you can store a memory. And maybe that memory can influence not necessarily what a gene does to produce a protein, but maybe changing a base can influence how much of the protein that gene produces. So maybe by changing a base near the gene, you still allow the gene to do what it's supposed to do, but it doesn't do it as efficiently, or maybe it does it more efficiently. Right? So now if you have a gene that's producing more of a protein, right, then that can produce more of a catalyst, for example, which can cause another neurotransmitter to modulate and fire more or fire less. Okay. So it's possible that DNA actually can store memories. We don't know. I'm not saying it does. I'm just saying it could. If it did, how could that look? So if we know that DNA sequences, if you look at DNA, it's 2 strands of DNA with the bases connected to each other. So if this was an A, and this was a T, then it could be connected like that. And by telling you the sequence on one side, you know the sequence on the other side because the A is always with a T, and the C is always with a G. So if I had this sequence, so here are my, so now instead of having zeros and ones, I have A, C, G's, and T's. This is called a quaternary system. Has anybody heard of a base 10 system? Base 10, 0, 1, 2, 3, 4, 5, 6, 7, 8, 9. I ran out of single digit numbers. I got to add a digit 1. 0, 1, 2, 3, 4, 5, 6, 7, 8, 9. I ran out again. I got to put a 2. Right? Binary is the same, except I only have zeros and ones. 0, 1, I ran out of digits. I got to put a 1. 0, 1 again. I ran out of digits over here. I get another digit over here, right? So there I can put a 1. So basically, just like we count in binary and we count in base 10, we have to also count in hexadecimal, which you're probably more familiar with, because that's pretty common in computer science. And here, this is called base 4. It means I have, I put 1, 2, 3, 4. You could also do 0, 1, 2, 3. But basically, if I was counting in 1, 2, 3, 4, then I would have 1, 2, 3, 4, ran out of digits. I got to put a 1 here. 1, 1, 1, 2, 1, 3, 1, 4. It's not 14 anymore the way you think about it. It looks like 14, right? After 1, 4 is what?
2, 1.
2, 1, 2, 2, 2, 3, 2, 4. So if in a test, an exam that's going to come up on April 1st, I were to say something like, in the quaternary system, here's this number, and that number were to look like, 14, and I would say what number comes after 14. I know no one in here is going to write down 1, 5. I want you to remember this. The reason I want you to remember this, not to become experts in quaternary and coding, but because as we think about how we want to store information, it may be that because the human system we know has memory in DNA, may not be the same memory that remembers language or the same memory how, that remembers what someone looks like, but we know for sure DNA is memory because it remembers how to make the cell, remembers how to make the cell function. It remembers how to make things that give you the color in your hair and the color in your eyes. Right? So we know that it is the genetic memory of who you are as a person. And it works in this system. When you get to RNA, you'll actually see that RNA works a little bit. It works the same in the sense that there's 1, 2, 3, 4, but there's words in RNA, and those words are limited to 3 spaces. So in RNA, you still use this language, but now you have 64 different words that you can make up. How do you make up 64 different words where you just do the same counting? I just did 0, 0. I'm sorry. If I'm using 1, 2, 3, 4, I shouldn't have done this. Probably do it 0 on the test. Whatever it is, I'll explain it to you. But for now, we're going to do 1, 2, 3, 4. Right? We would have 1, 1, 2, 3, 4, 2, 1, 2, 3, 4, 3, 1, 2, 3, 4. And then we go to the next one. We still can do that. 4, 1, 2, 3, 4. Then we get to start all over again with moving this 1 to 2, and then going up 1, 2, 3. And so you can get 64 combinations of different things. Okay, so in this example, I'm doing is saying, okay, if an A is 1, and a C is 2, and a G is 3, and a T is 4, then this sequence, which is half of my DNA, is 1341123. What if I wanted to write this in binary? What would it look like? Right? So in binary, to do it, I have to use 2 digits. So I can say 00 is A, 01 is C, 10 is G, 11 is T. Right? So my sequence of A, G, T, A, C, G, T, T would now be this sequence here, of zeros and ones. So remember, because zeros and ones can be used in a computer to store anything, right? If you have a picture, a movie, your messages, then you could envision creating a strand of DNA, right, that had your picture in it. Basically, if you just said, look, I'm going to create this cool looking 3D printed strand of DNA, and it's going to be a picture of me because I'm going to decide to encode the sequence of this particular DNA, not like any human I've ever seen, but just in a way that converts these zeros and ones into that. And that's the same if we were going to do this in hexadecimal. So this is just showing this in hexadecimal, which would be 2C1B. So we have a lot of options in terms of how we want to represent memory, and it may be that if we want to model how memory is stored in different biological systems, that we may not want to use binary when we're thinking about it. We may not want to use base 10. We may, so we may want to use other numbering systems. Genes are made up of DNA, so if you have, in a chromosome, 100 million base pairs that line up to make this DNA, remember that only about 1% of it has actually been identified to actually be genes. And so what does that mean? That means that most of your DNA is what they call its non-coding DNA. And some portions of your DNA, which vary in length, will be your gene. I've already mentioned this. There's about 25,000 human genes that have been identified within your DNA. Expression, I talked about just a little while ago. The genes create protein, basically, and I'm going to show you how they do that. How much protein they create, and when they're creating protein is what it means when they say is that the gene's being expressed. It just means that the gene's not just tied up in the DNA where it can't do anything, but it's opening up and actually doing something. So how much it's expressed is what I talked about before. It's something that can be modulated. And this talks about how, I'm not going to go through this here, but please go through it later. This talks about different ways that the non-coding DNA, if it were to store memory, like actually the memories of language and memories of what your friends look like, if it were to do that, how it could do that by having some of the DNA base combinations change a little bit, mutate, what they call, near the gene, so the gene would still do what it's supposed to be doing, but it may not do it as efficiently, or it may do it more efficiently. Okay. So I don't know if that's clear what I mean by regulation, but think about it like a knob. Okay. If what the gene is supposed to do is play a song at a certain volume, and then you change some of the coding near the DNA, and it makes near the gene, and it makes the gene more efficient, then it would be like turning the volume up. It makes it less efficient, it would be like turning the volume down. So obviously, if you can modulate, like by turning something up and down, then you can communicate that information that you stored. So what the gene actually does, and so this is a strand of DNA here. This would be like, you can see here, there's A, C, G, T. We have it color coded here. So here's your sequence going down one side. Sequence is the same on the other side. When it opens up, the gene now, the part of the DNA that's going to do something, can interact with something that's called RNA. Basically. And it's going to essentially create this RNA, which looks a lot like DNA, except it's not. Okay. It does follow the sequence. So it mirrors the sequence. And by the way, because this DNA is complementary, if you have, I mentioned this before, but it's really important. It splits apart, even though the sequence isn't the same because it's complementary, you know what the other sequence is by looking at inside. The A is always with the T, the C is always with the G. When you do the RNA, there is no T, but actually, RNA stands for ribonucleic acid, so it's still nucleic acid. You still actually have your A, C, and G. It's just instead of the thymine, you have a different molecule. It's called uracil. So this DNA can now create this RNA, and then the DNA, the gene can close back up again. This RNA molecule that now basically has read a little bit of the copy of that gene can now move through the cell and get to a ribosome. Why do you want to get to a ribosome? Beside the fact that we're almost done with the class. Okay, you want to get to the ribosome because the ribosome makes proteins. Proteins are life, basically. Like you are proteins. And proteins are what kind of proteins, how many proteins to make, which cells should have what proteins, all that's decided by the DNA and the cell environment that the DNA is in. So this DNA created this RNA. The RNA basically read out like the sequence of that part of the gene. It got to this ribosome, which is here. So this mirrors the sequence of the DNA, and I mentioned that we use 3 letters. That gives you 64 combinations. Each one of these words, if you will, 3-letter words, corresponds to different amino acids. Not each one, because there's less amino acids than there are 20 of these. But basically, these amino acids are attached to 3 RNA characters, if you will, and it reads the sequence, and this ribosome magically makes a chain of amino acids, and that's what protein is, a bunch of amino acids in different combinations. So it makes proteins. Proteins do things. Remember the proteins that go into the membrane and their channels? That's one example of protein. So that's what allows the cell to receive and respond to a neurotransmitter, open up, send electrical signals. So you can see how DNA, how RNA, how all these things can change the behavior of the cell and modulate what's going on electrically. That's really the highest level point what I'm trying to get across in the class. Lots of ways that your brain can store information. So, it's a lot of words here, but this is actually readable. I put this together right before I came in here, so I didn't want you to forget. The brain cells. We've decided that the brain is where most of us is going on. These brain cells, there are neurons and there's glia, right? There's over 100 billion of all of these combined. They have a nucleus. The nucleus has chromosomes. Chromosomes are comprised of DNA. The DNA is comprised of parts of the DNA have genes. The genes encode messenger RNA. Messenger RNA are consumed by the ribosomes to make proteins. Proteins are actually what make us biological entities from our cells. And memories can be formed by any or all of the cell parts, starting from the DNA to the proteins the dendrites and the axons, to the concentrations of proteins in the cell, the composition of it, to its connections, or even its physical shape. Okay. In cellular memory, it could be digital, analog, or both. I didn't really talk about the analog part. But when you look at how signals go through a dendrite, it's possible that that can actually shape the form of the signal that comes out the other side. Remember our analog memory discussions, it's shaping that signal so that the action potential has some distribution that's associated with it that has some additional information inside. Okay, so closing thoughts. Sorry, I ran over. Basically, senses all worked with different types of receptors. Remember that? So we had our senses, we had our cells, the receptor would get activated. It would send a signal somehow, somewhere, that we would then interpret in some way. So all that memory has to do, really, is create something, remember, and then create a stimulus that's similar to the stimulus that the original receptors, which were also proteins, created in the cells. So it's not that, to me, it's not actually that complicated to think, well, if receptors in cells could transduce this original stimulus to send electrical signals that my brain could see or touch or feel or smell, then it doesn't seem that magical to me, actually, that those very same things could be configured in a way to do it again in some way. And especially since I don't see things when I have my eyes closed in my mind as vividly as I see it when I have my eyes open. So next time, because you allowed me to go a little over and get done with memory, next time we're going to finally get to go into perception. And then the homework, like I said, it's probably not going to be available till Thursday, but it's going to be more straightforward going through the topics we've discussed, just to make sure that you understand the important parts. Okay, thanks.